The Nobel Assembly at Karolinska Institutet has today decided to award the 2017 Nobel Prize in Physiology or Medicine jointly to Jeffrey C. Hall, Майклу Розбашу, and Michael W. Young for their discoveries of molecular mechanisms controlling the circadian rhythm. Here are the three laureates. Jeffrey Hall was born in New York and performed his seminal work at Brandeis University. He's now retired and lives in Cambridge in Boston. Майклу Розбашу was born in Oklahoma City and performed his prize-winning studies also at Brandeis University, where he's still on the faculty. And finally, Michael Young was born in Miami and did his work at Rockefeller University in New York, where he also remains on the faculty. I will now describe the discoveries behind this year's award. So this year's prize concerns the circadian rhythm. The word circadian originates from the Latin word circa, meaning around, and dies, meaning day. But in a way, it is actually about astronomy. Ever since the emergence of life on Earth, about 4 billion years ago, evolving life forms had to adapt to the rotation of our planet. This ability to prepare for the regular daily fluctuations is crucial for all life forms. But how is this possible? This year's Nobel laureates have been studying this fundamental problem and solved the mystery of how an inner clock in most of our cells in our bodies can anticipate daily fluctuations between night and day to optimize our behavior and physiology. This existence of an inner clock was first described already in the 18th century. The French astronomer de Meuron studied the mimosa plant that opens its leaves towards the sun during day but closes at dusk. De Meuron asked what would happen if the plant was exposed to constant darkness. And he found that even then, for several days, the leaves continued to follow their regular 24-hour daily rhythm. In the 20th century, it was established that also other plants and animals, including rodents and humans, have their own biological clocks following the daily circadian rhythm. Studies of the clock in fruit flies show that many different behaviors are controlled by the clock. Fruit flies are really excellent model organisms for studies of genetics, and in the 1970s, a mutation that affected the fly's daily rhythm was identified. The mutated locus was called period. But how did the mutation affect the internal clock? This year's laureates, they wish to peek inside the clock and figure out how it actually works. A first breakthrough came in 1984 when Jeff Hall, in collaboration with Майклу Розбашу and Michael Young working independently, were able to isolate the period gene. Now the scientists could begin to disentangle the cellular wheels and cogs of a clock machinery. Jeff Hall and Michael Rozbašu showed that the period protein itself was cycling and peaked during night and was low during day. In 1990, they showed that the period mRNA also oscillated, as shown on this slide, and accumulated several hours before the period protein. These and other important observations led to a brilliant idea for how the clock works by a feedback control mechanism. The idea was shown to be correct, and in the next few slides, I will show how it actually works. When the period gene is active, period mRNA is made. The period mRNA is transported to the cell's cytoplasm, where period protein is made. When period protein accumulates, the protein moves into the cell's nucleus, where it blocks the activity of its own gene. When the gene is blocked, no more RNA can be made. period protein levels will decrease, and the gene will no longer be blocked. And the production of mRNA can start all over again, and the cycle can continue. But it remained unclear how the period protein could accumulate in the nucleus to inhibit the gene. This problem was solved when Майклу Янгу discovered a second gene called timeless that is also critical for the clock. This is shown in this experiment. The top diagram shows how period mRNA is cycling over several days in normal flies. The bottom diagram shows that the cycling was disrupted when the timeless gene had been inactivated. But how did the timeless protein affect the period gene? Additional experiments show that timeless protein bound to the period protein, and together they entered the cell nucleus where they could inhibit the period gene. It was also critical to understand how the speed of the oscillations can be controlled. Майклу Янгу identified yet another protein called double time, or DBT. This protein was shown to slow down the accumulation of the period protein, which explained how the oscillation can be adjusted to more closely match a 24-hour cycle. Jeffrey Hollu and Майклу Розбашу showed that two additional proteins, clock and cycle, activated the period gene in fruit flies. So, with all these groundbreaking discoveries by the three laureates, an elegant feedback control mechanism for the biological clock had finally been unraveled. We now recognize that biological clocks function by these same principles in other animals, including in ourselves, in humans. Most of our cells in our bodies have these clocks. Our inner clock adapts our physiology to the dramatically different phases of the day and regulates many critical functions such as behavior, hormone levels, sleep, body temperature, and metabolism. We do not feel well when there is a temporary mismatch between our external environment and the internal biological clock, for example, when we experience jet lag. Studies have also indicated that chronic misalignment between our lifestyles and the clock is associated with increased risk for various diseases. Since the paradigm-shifting discoveries by Hall, Rozbašu, and Young, circadian biology has developed into a highly dynamic research field with vast implications for our health and well-being.
We don't know of any drastic disruptions in humans of the clock. There are, of course, animal experiments, and if you disrupt the clock in animals, you will get effects on metabolism and on various physiological parameters. But in humans, it's not really possible to do those kinds of experiments.
Нужен анонимный доступ к любому сайту? Хотите общаться, не раскрывая свою личность? Планируете рекламу ВКонтакте, Одноклассниках и других социальных сетях? Соцсекрет предлагает легкое решение этих задач. Мы продаем чистые, созданные с нуля аккаунты для любых сайтов. Покупая аккаунт, вы получаете уникальный логин и пароль без необходимости вводить собственный номер телефона и где-либо регистрироваться. 100% гарантия работоспособности. Стоимость анонимного аккаунта всего лишь 30 рублей. Адрес сайта соцсекрет.ру. Ссылка в описании.